So let's see. With synthetic biology, I think, yeah, it would help if I'm plugged in here. That would be a good idea. Okay, let's stop sharing here for a second because now it was saying my speaker was weird, but it should be fine. All right, let's try that again. So synthetic biology has a bright future. And lots of exciting opportunities. Businesses are popping up, looking at how we can use microbes. So in the realm of synthetic biology, microbes are the workhorse, although I have seen them working with plants as well. I've gone to some plant biology conferences where there are symposia sessions on synthetic biology plants, but it really did start out in the realm of microbiology, and it seems like that's where it's really going strong, mainly because microbes are pretty easy to work with. They're cheap to work with for the most part, easy to grow in small spaces, and are pretty open to being manipulated in the sense of their genetic manipulation in particular. So with synthetic biology, we're looking at really some interesting tinkering going on, creating new pathways. I went to one synthetic biology conference a few years ago, and I was just blown away at the advances they had made since the previous 2 years before that. They're making advances in leaps and bounds, and it's hard to keep up with all the stuff going on in this realm of research and application. But looking at, and I had this idea of consortia popping up everywhere. They're really starting to get excited about this idea because they realize that in many cases, working with multiple different species together can get them to accomplish something that they want. And then if they just tweak it a little bit, they can really maximize what they're trying to accomplish. And of course, make a profit off it too. These are businesses, they're entrepreneurs, they're trying to get this to market, but not go bankrupt. So the consortia idea is gaining, and now we have the technology where that can be looked at and it can be studied. And they're really looking at how can we use this there's a lot of cancer research going on use of these microbes to either be therapeutic agents or maybe be, like the idea of having them deliver a payload of cancer drugs to the tumor. These types of ideas are gaining a lot of interest. They're even looking at quorum sensing ideas, how better to understand quorum sensing, that they can use that in this synthetic biology process. So we're saying we apply to consortia. So again, work with mixtures of microbes. This idea of interspecies communication, quorum sensing between species is something that they recognize and that they need to understand that better and hopefully find ways to work with it to get what they want accomplished. And so more and more of those kinds of papers I'm seeing published. They're certainly looking at antibiotics, but also virulence factors. Some people are in the marine ecosystem looking at coral reef systems, but a lot of people are really focused in on human diseases and disorders. These are just snippets of published papers. Probiotics, using microbial biosensors. Biofilms is a hot topic. How do we manipulate biofilms the way we want them to be? A lot of the times we don't want biofilms. Other times they do. It depends on the situation. Antivirals, plastics, and that's a big one. Boy, is that gaining a lot of interest. rightly so, in the idea of breaking down plastics in our environment. And have you all heard about PFAS? 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 That's a real hot topic right now. Micronaut, the local company that Joe Lane and Ryan Iacovaci have founded, they're very interested in using mushrooms as remediation for PFAS. And so they're working hard on that, trying to get grant funding to pursue that. I just saw a blurb in, did I tell you this? The ocean currents and the ocean spray as the ocean waves crash onto shore. They're taking the aerosols from that and they're finding very high levels of PFAS. Isn't that a lovely idea? And so now they're saying that PFAS is accumulating on the surface of oceans. It tends to float, not sink. And then when it comes to shore and splashes and you have that aerosol kick up like that, they can document that, you know, people, animals, organisms in that area are getting exposed. So it's this never ending plastic issue that needs multiple interdisciplinary and lots of different ways of looking at it to try to solve this problem. They're looking at crops too. How do we use microbes and so this is another idea, sustainable agriculture, big idea right now. And also not only do agricultural in a sustainable method, but how do we feed all these people? What is it, 50 billion by, what is it, 10 billion by 2050? Is that what it's supposed to be? By the year 2050, it could be 10 billion people on the planet. If you talk to people who are in the agricultural industry, they will go, I'm not sure how we're going to do that. And so it's a little scary when I go to these plant biology conferences where they admit that if you just rely on our conventional agriculture system, we don't have enough land to feed that many people. And that's why, that's one of the reasons, and this idea of sustainable agriculture is a whole other issue. How do we do it sustainably so we don't keep wrecking the environment? And so the idea of indoor agriculture, that's one of the reasons why I got involved in designing that program here, is that we're running out of land. We got to think of other ways to grow crops. And so using microbes in that process, I think, is definitely a way to go, and is in the initial stages of research, but definitely, you know, they're looking at how can we go back to that. And they're engineering microbes to fix nitrogen, for example, and getting more crops to interact with these microbes in a synergistic way. And that will go a long way toward, you know, less nitrogen fertilization going on, less pollution of that into the soil and waterways. So lots of exciting ideas, looking at microbes and getting the microbes to feed the plants essentially, as well as protect the plants, be biocontrol agents and protect plants. Pharmaceutically, of course, they're looking at, there's a lot of research going on. I highlighted that one, the cannabinoids. They're looking at plant alkaloids and cannabinoids. And so they're, you know, that's a huge market. There's a lot of interest in it. And so they're asking the question, you know, is there another way to provide these medicinal products than growing the cannabis plant? Is there something else we can do? And so they've actually done it in yeast, which I thought was very, very interesting. So complete biosynthesis of cannabinoids and their unnatural analogs of yeast. So they're taking yeast and using synthetic biology techniques to get it to make these cannabinoids. Working with yeast, like I said, much easier than working with a bunch of hemp plants or cannabis plants. A lot less space, a lot reduction in cost and labor, as well as supplies. So there's a lot of attractive reasons to switch into using microbes for drug manufacturing, essentially. So again, this is coming a different review paper. Looking at how can we do this outside of using this. So I think you're gonna see more of that. Has anyone heard about this yet? Anyone maybe in the plant medicinal chem program? So it's gonna show up more because I think there was not this conference that just happened, but a couple years ago, there was a speaker where they were looking at this idea. And so it's already in the works. They just need to see if they can scale it up essentially. Here's one that has a lot of impact: sugar without calories. Stevia. Stevia, some people say. I suppose that's probably more the correct way to pronounce it. I don't know why, but I know it's stevia. Stevia. So that comes from plants. I've grown it in the greenhouse, and if you take a leaf from that plant and just chew on it, oh my gosh, the sweet factor is about knocks you over. It is incredibly how sweet leaves are, and that's where they extract it from. And so it's a safe alternative to sugar because it's calorie free from our perspective and physiology. But it can create a bitter taste when you extract it from the leaves. Have you ever tasted stevia that you willingly knew you were tasting? It's in a lot of different products, but usually people your age, you can tell when you're tasting it because your taste buds are pretty fresh. Mine. And so you taste the bitterness. My sons I can't sneak stevia into their food. They know. They're like, there's stevia. And so they're trying to figure out a way to make it bitter. This company called Evolva, they are trying to do that. And I keep looking for them to be on the market. I haven't seen it yet, but it's not there. But this is huge. Can you imagine the profit they're going to make if they can get this mapped out? Here's another one. Beer with caffeine. Kind of seems odd. I'm not sure I would pick that, but they are manipulating yeast using light as part of the manipulation. They actually will turn different colors of light onto the yeast to get different pathways to pick up. So they've done this through synthetic biology. And so they're getting it so that if they do all the controls the right way, they can actually get the caffeine production ramped up. Limonene is they can choose to get that ramped up, and they'll have a lemony flavor to the beer. And then some of these other pathways create anti-cancer effects. So drinking beer with anti-cancer properties is a pretty novel idea. Have you heard about any of this? So it's new. I mean, it's early days. Sweeter beer, but I thought this was a nice graphic, just trying to show that the ability of manipulation that they have if they use the synthetic biology processes to engineer these different pathways, but then to also engineer tight controls on them. So just with a simple flip of the light, they can get different pathways turned on, different products. Really, really interesting. Here's another one with yeast again. Milk from yeast. So this has a lot of people really excited because the normal production for cow's milk is controversial for many people, and growing cows is expensive, and greenhouse gas emissions and all of that. By the way, I think I told you this, probably don't remember, but we were looking at the rumen of cows. It's not the cow themselves that gives off methane gas, it's the microbes in the cow. So the cow gets the blame. People say cows cause greenhouse gas. Well, yeah, but it's really microbes inside that are doing it. But anyway, it takes about 1,000 liters of water to produce 1 liter of milk. Moo free. Moo free is the company, the product that they're trying to animal-free milk. Would you drink it? I think it'd be worth trying. I don't know. I looked for this earlier in the semester. I could not find anything yet on how far they made it through the all the testing. Last I heard, they still had a couple of end pathways they needed to tinker with. So they're bioengineering yeast to produce authentic milk proteins, which will give it the same taste and nutrition as regular milk. They say they already match 97% of the taste of milk. So they're trying to create something that's essentially identical in terms of nutrition and taste. In terms of so that has big application. And then of course they're looking at how do we use this for cancer. Here's a kind of a biosensor idea where you eat yogurt and the microbes in the yogurt are bioengineered so that they can pick up on tumor cells in the body and it will actually turn color. So you essentially pee into a cup or have a clean toilet bowl to pee into, and you can tell by the urine color. So a biosensor. Have you seen the cat litter idea where they do this with cat litter? It's not quite the same thing, but it's a color changing thing in cat litter to know if your cat is healthy or sick. But here they're doing it where the microbes are bioengineered to react to the tumor cells, and then a very simple visual color shift. I mean, that's brilliant. That's a great idea. And so there's these types of ideas on and on and on in the realm of synthetic biology. And I know in your lifetime, you're going to be able to take advantage of some of this in terms of improvements in human diagnostic tools, therapeutic tools, just overall increasing human and other animal health. So keep your ear out and eyes out for that. There's more to come on that for sure. I put this quote in there as well. This is Edward O. Wilson, who is a very famous biologist and naturalist, and he is not a microbiologist. But he said if he could do it all over again, he would be a microbial ecologist. So after learning more about microbes, he became so enamored with all they have to offer and how amazing. He studies, the big fuzzy animals and thinks about things like that. But yeah. So I thought that was a nice nod to how awesome microbes are. Even somebody who doesn't study microbes is willing to admit that, and someone that famous. So I had to put that quote in there. Now, this quote is from a microbiologist, so it's a little biased, but it's true. We live in the age of bacteria. And so I think so many people don't appreciate that microbes were here first, and then we evolved within their world, essentially. And so we are constantly interacting and surrounded by, or, when you look at the evolutionary tree of life, it was microbes that started it all off. And so this idea that we evolved into their world, but a lot of people don't look at it that way when they think about evolution. But hopefully, after this class, you can think a little differently about that whole idea, and that hopefully next week you'll say you survived. So that's the goal. Let's survive this. Let's get through it. All right, so we need to think about the final exam. It's going to start with viruses, right? Because we ended with fungi in the last one. We need to think about viruses, the human microbe interactions, a short stint into epidemiology and food microbiology, and then this last topic of applied microbiology. So 5 different topics and the associated readings will be the new material. far as cumulative, I know that the two questions I asked on this previous essay exam will also be part of the cumulative. Remember for that, I said anything cumulative will show up more in the multiple choice portion. That doesn't necessarily mean it'll be a multiple choice question. It might be a matching question or something like that. But let me pull up those two questions. So going back, there was a question on quorum sensing with B serious, saying that you are a B serious and you ended up getting eaten by a human, you're in the gastrointestinal tract. Explain the process of quorum sensing start to finish. So with gram positives, that's the two component signal transduction system. Whoops, hang on just a sec. And for the gram positives, okay. So from start to finish, I needed you to explain the idea that as serious doing quorum sensing, you would be making these peptides and excreting them out, and somehow getting across the point that you would be waiting for these signals from other microbes as well. You'd be monitoring that. And essentially, when you get enough microbes all doing this in the same area, there's going to be a threshold level where that kind of tipping point where now you get the quorum sensing signal into the sensor kinase protein. Once that happens, the sensor kinase will pass a phosphate through to the response regulator in the cell. And that's the signal to the response regulator to go find the DNA. And it's fair to just say, and alter gene expression. So that was for part 8 on the essay from start to finish. Questions on that idea? Part B was asking about why would you do quorum sensing? Why didn't you just immediately, you're a human pathogen, I stated in the question, you're a human B serious pathogen. So why didn't you just behave like a pathogen immediately? Most of you did quite well on part B, where you were explaining that it wouldn't be a good idea once if it was just you or a handful of you getting swallowed. You don't want to try to be pathogenic when there's too few of you. You want to wait, assess, figure out when you have an army. So you always have to be doing this quorum sensing to be able to assess population levels. Once you have enough, once you have that army, then you attack, and you'll have a much higher chance of success. So something of that answer, something like that is what I'm looking for. And most of you did quite well. But the first part was a little rough. So that's why I'd like to ask about it again. Quorum sensing is a thing. You're going to see a lot more of that as being used for different applications in your life that will affect you, hopefully in very good ways. But I'd like you to have a better understanding of that. Okay. Do gram negatives use peptides? No. So it's going to be fair game for me to ask a little bit about gram negatives as well. So be prepared for that. So gram negatives don't use peptides as their chemical signal. What do they use instead? Yeah, homoserine lactones. So pull out those notes as well, and think about So some people call them AHLs, but it's homoserine lactones. A little simpler process because once there's enough of that chemical signal, which means there's enough cells in the area, you'll get diffusion into the cells, and they'll find the regulatory protein aligned with that. That whole complex will then interact with the DNA and alter gene expression. So a little bit more straightforward than what we think about with the gram-positives. Questions on quorum sensing? The other question was on photosynthesis. Photosystem 1 and photosystem 2. So when we covered photosynthesis, I'm gonna hang on just a second. When we covered photosynthesis, we thought about how plants do it and how cyanobacteria do it, and they have two photosystems. But then we learned that there's some microbes that just have photosystem 2 and work it just with that, or they just have photosystem 1. And so I asked for one similarity, I think, between those two. And when you look at what's going on, the big similarity there is that through an electron transport chain process, they're able to generate. They both can do that, whether they're using photosystem 2 or photosystem 1. So here's photosystem 1. And of course, the artist did not put in there the ATP, but I have it in the notes. And then photosystem 2. So that's what I was looking for the similarity, the idea of an electron transport chain linked to proton pumping that gets you some ATP. They're different. One huge difference is that with photosystem 2, they have no way to directly make the NADH or the NADPH. That is also a huge part of photosynthesis, one of the goals. When you think about it from how plants do it, cyanobacteria, photosystem 2, those microbes that you spoke to, they have no direct way to make NADH. However, photosystem 1, they can do that. So that's a huge difference. Another huge difference is that photosystem 1 uses a linear flow of electrons. Okay, it starts with an external source like hydrogen sulfide, we mentioned that, and ends up at a plus. So it's linear flow. Whereas photosystem 2, they don't have that. They don't have that external electron source. They actually use the bacterial chlorophyll in the reaction center to put electron on that. And so it's a circular flow. So that was another huge difference. So I'll be asking the basics of photosystem 1 and photosystem 2. Again, it'll be in the multiple choice portion. So just make sure to brush up on that. So if you go into EDUCAT and look at your essay, I have the answers there. They're what I was looking for. So that's a good study tool. Okay. Questions? Let's think about the other topics. I'm going to pull up the viruses. So there was 2 parts to the virus topic, 2 slide sets. All right, so this is the first slide set. What kinds of questions do you have about viruses? Anything confusing, or if you want to know anything about it for like the exam? You looked at kind of their anatomy, I guess. You could break it down into their anatomy, which I'll be asking about, and then a little bit about classification, a little bit about how they reproduce, and well, how they replicate is a better way to say it. And all the way from they got to make nucleic acids, they have to make their own proteins. So we looked at some of the different strategies. It really does depend on what type of virus they are, if they're an RNA or DNA, and then are they single-stranded or double-stranded. That makes a difference on how they will go through replication. We looked at how to study viruses. Can't really look at them directly unless you have an electron microscope, but you can do plaque assays and things like that. And you can work with, phages that attack bacteria. You can work with viruses that infect animals. You can do plaque assays with both. Got into the replication ideas, a little bit of how bacteria try to fight back against viral invasion and getting infected. Any questions on these topics? What about the second part? That was looking at ecology of viruses, focusing on more on the environmental, I guess, ideas, not so much the pathogenic viruses. Looking at the ways that they interact with our Earth and us in more, I guess, beneficial ways. Did look a little bit at oncogenic viruses, but remember, I just want you to know the list, really. I'm not going to ask details of each one. That's more just for your own information. So really, after you get past slide, in my slide set it's slide 26. So when you get into the different slides showing different, like Hep B, papilloma, I won't ask details on those. But knowing the basics, like slide 26, where it's getting into the like how does it trigger cancer idea, the concept of that, like how it works in general, is fair game. But I won't go into ask details on each virus that's in the list here. Any questions on the viruses? What about human microbes? That was a big slide set. different body parts. We kind of zoomed in on the gastrointestinal tract for most of this, but we did cover the other body parts first. So we spent a fair bit of time on this study with the cute little mice here. So being able to talk about that one is a good idea. Like how they set up the experiment with the identical twins, with the mice. So I would like you to think about that experiment. That was a groundbreaking experiment that really kind of broke open this whole concept of how impactful our microbiome can be, and that it can actually transmit into across, species here. In this case, it was with the concept of obesity. Questions on the human microbiome? We all tapped out. Second slide set, we got into some of the more details on what are they producing, the short chain fatty acids. Is a big one that they're really excited about. Some are good, some not so good. Ways that they impact our brain, our behavior, our mood, the gut brain, inflammation, huge concept right now. Any questions on this stuff? Well, what about epidemiology? Anything there? Epidemiology, where are you? Pretty short slide set. Yeah, so that there's, some definite, like there's some definitions in here, some concepts to think about, but overall, pretty short slide set. Food microbiology. Remember, I won't really ask you anything about food spoilage. It's more focusing on the food contamination idea. And just remember, when you eat plants, you're eating plenty of microbes. Even if you wash your plants off, you're getting microbes. Most of those are just fine, but they are worried about some of the human pathogens that can be lurking, especially if people who grow the crops aren't paying attention to some of the issues in the field of contamination that can occur. Hey, did you hear there's a recall on ground meat for E. coli? Out east, I think, is where it started. I just checked the CDC, it hasn't showed up there yet, but I've seen numerous news alerts on my micro stuff. And so, yeah, they're issuing warnings. And the problem is the ground beef got sold to restaurants and supermarkets. So it's kind of a double whammy. But yeah, E. coli 0157H7 is showing up in ground beef made for, for hamburgers. So you might not want to eat hamburgers for a little bit. But anyway, so some of the ways in which contamination can occur, some of our human activities that are making it harder for epidemiologists. Boy, that's a tough job. Must be, if you love solving mysteries, boy, that's a good job to get into. Differences between food poisoning and the idea of toxin production with food poisoning, or if you are doing more of a foodborne infection where the microbe has to get in you, has to multiply, takes a little bit longer, but then eventually they start causing problems. Viruses win in sheer numbers. And then with the food preservation, just the basics. These tables are more for your just general interest. I'm not going to ask anything on these tables. Questions on food microbiology? I sense exhaustion. Exhaustion and low energy. I get it. Well, the last topic is applied, what we just covered on that slide set, and that one isn't that big either. So the biggest topic is human microbe. I'm guessing most points are going to come from that. It's also really exciting area of research right now, lots of amazing discoveries. So human microbe, there's going to be a fair bit of more points coming from that topic than any other, I'm predicting. The next topic in terms of point value will probably be viruses. And then the other ones, I haven't mapped that out. I don't have a final version yet. As I get closer to a final version, I can give you an update and kind of give you, especially for the final exam, I tend to rank it like this, where you know where the bulk points are coming from, so you can strategize. Because I know final exam week can be pretty tough. So I'll try to get that data to you when I know better on that. This applied slide set, again, not terribly long. So the basics on this, there's definitely some ideas you need to learn, but like I said, some of these tables that I have in here, I might ask you, like, give me an example, but you don't have to like memorize the table. It's more like be familiar with something. So that kind of like if you're at a party, you can like say something interesting from one of these topics. Any final, just general questions about the final exam? Anything that you want to know? Any questions? It's going to be here again in this room. It's full 2 hours. Same idea, multiple choice versus a separate one on essay. If I need to send you out another announcement about the cumulative portion. So the quorum sensing and photosynthesis for sure. I need to double check the other 2 exams to make sure that there isn't something else that I want to grab. But I'll let you know exactly what it is if I do decide another question. So essay will be new stuff. It'll be pretty similar to the previous exam. It might be just a tad longer with a few more, like the human microbe. There's going to be a good chunk of points on that, so that tends to extend it a little, and then a few cumulative. So probably a little bit longer, but certainly 2 hours is more. It'll be more than enough time to get it accomplished. For sure 2. I don't think I'll go above 4. I can let you know when get a solid draft. It's also time crunch on faculty, on the faculty side, because they demand our grades be done the following Tuesday. And that can be very hard to accomplish. So I have to be careful on how many manual grading things I have to do. The other thing I want to remind you is the evaluations for the course. Getting into EDUCAT and clicking on that link and doing the evaluations both for lecture and for lab. Right? And that's important data. We actually use that data. If we pull that into our portfolios and we have to report out on that, and it just helps us run our department better. We can figure out how we need to shift things, what to do better. So it is valuable information for me and for all of us. And so I really do ask that you take a few minutes to provide your answers for that. Okay, both for lecture and class. There was something else. What was? Oh, do you need extra time? Let's see. Today's Wednesday, so I guess after tomorrow you'll know better, right? On if you need to come in again in lab, or if tomorrow is enough. So work with Duncan and Jackie. And they did say they could be available. So hopefully something could get worked out if you need to come in one more time beyond Thursday. Okay. So communicate with them. All right. We'll stop there.